That's very important. I think today we are going to have Dennis Volkl. I met him in ISMB and ECCB 2025 in Liverpool, and I found his um, research is very interesting, where he used the box sequence data to do this single cell type deconvolution. So we are looking forward to your talk, and the stage is yours.
Sorry, wait. You're on mute. Huh? Mute.
Go ahead, Dennis.
Okay, good. Well then, thank you for inviting me. Still remember our talk at my poster. Was pretty amazed that you invited me here. So I'm Dennis, and I'm a PhD student at the computational biology unit here at the University of Bergen in Norway. And today I'm going to present you about my deconvolution model HIDE. There are a lot of different deconvolution models out there. And what I proposed differently than the others is that I use a hierarchical relationship between various cell types to deconvolve the cell type proportions in bulk RNA sequencing samples. Well, before I want to dig into the algorithm, just let me briefly explain you why we are even interested in the cellular composition of tissues. The research over the past decade has shown that cancer cells are able to influence their surrounding. This means they not only spread uncontrolled, but they are able to shift the cellular composition around it. For example, I've read a paper recently where they showed that fibroblasts can be arranged around the tumor to some kind protected. And this so-called tumor microenvironment then influences response to therapies, for example, or metastatic behavior and other things. And therefore, especially in cancer research, knowledge about these cellular proportions and how they are present in the tissue is very important. But how can we even measure this? Well, the older method and I think most common method is by bulk RNA sequencing methods. Here we take a whole sample and dissolve it and then measure the gene expression of the sample. If you think of it from a more mathematical perspective, it's just like measuring the gene expression as a weighted sum of all of the present cell types. As you might think now, the true cellular composition of this still remains unknown. Another newer method then is single cell RNA sequencing, where we, instead of measuring the whole gene expression of the whole tissue, we measure the gene expression of each single cell. This, however, makes it necessary to later annotate each sample or each cell measured with tools like Seurat to get the cell type label. Well, obviously, you might now wonder why don't we just use single cell RNA sequencing? Because we measure each cell single, we know each label, at least if the annotation worked. However, the single cell RNA sequencing is pretty expensive and has scalability problems. While with bulk RNA sequencing, we can perform high throughput studies with hundreds of patients at once. You have a high cost factor in single cell RNA sequencing. As it's a newer method, you need more chemicals for each single cell, and you have an increased experimental effort. And also, on the other hand, bulk RNA sequencing is an older method. So there are many publicly available data sets around there, and it would be unfortunate not to use the information that has been measured over the past years. However, if we use bulk RNA sequencing, we still want to measure or to get an estimate of the cellular composition of the sample. And this can be done by using so-called deconvolution methods. And one specific type of deconvolution method is the so-called reference-based deconvolution. Just think of that you can describe a cell by an archetypical reference profile, meaning that we assume that each cell of the same type has some kind of consensus gene expression profile. Then we can describe the bulk as pictured in the figure included as a product of some weighting factor times this archetypical reference profile to resemble the bulk. This, on the other hand, still comes with some problems. First, you have closely related cell types like T regulatory cells and T follicular helper cells, which you can see on the UMAP plot on the bottom as pink and oranges. You see that this cluster is like highly intertwined, and this leads to high collinearity in the reference profile, and thus makes it pretty hard to estimate those cell types correctly. Furthermore, some cell types are not really abundant, but instead very seldom. So you have a very bad signal to noise ratio. This is also bad if you try to deconvolve them computationally. And as another problem, sometimes you're more interested in the major cell types, like you're only interested in how large the percentage of epithelial cells and immune cells is in a sample, but sometimes you're interested in specific small cell types like CD4 positive T cells or CD8 positive T cells. And if we now run the same algorithm on the same sample, but at two different resolutions, we can see that we have different results. For example, the coarsest grained layer predicts us that there are no T cells in the sample. However, if we go to a finer grained resolution, we could get in theory a result that shows that there are many CD4 positive T cells in the sample, which obviously doesn't make sense. The first three problems can be solved with multiple algorithms. For example, the one proposed by our group, which is called digital tissue deconvolution. Here we include learnable gene weights, meaning that we weigh genes if they are important for the deconvolution problem, and if those genes are not really important to distinguish between cell types, we weigh them down. Computationally, we can describe it as two different loss functions. The first loss function is by just solving a least squares problem for fixed gene weight. And in the second step, we learn those gene weights by maximizing correlation between the estimate and the ground truth. I've included you a plot here where you can show that this gene weighting enables us to find out about cell types. On the left-hand side of the plot, you see macrophage prediction without using gene weights, where with a correlation of about 0.0, we can't really estimate macrophage population. But on the right side, you see what happens if we include this factor. We can now really good predict macrophages with a correlation of about 83%. This still leaves us then with the problem of the inconsistency, which I mentioned earlier. And this is where I come in with my algorithm. We know that cells are related. So we know that T cells split up into CD4 positive and CD8 positive T cells. And each of these two cell types then have their respective subtypes. And we now want to use this information to get better predictions of the small types, but also enable the user or the biologist to get a prediction at each step in this hierarchy. Let me now briefly explain you how this model works. This model is a top-down approach, meaning that we first perform the deconvolution on the coarsest grained layer, meaning the major cell types like T cells, epithelial cells, myeloid cells. And then we use the knowledge from this better solvable problem to deconvolve the lower levels. The first step, therefore, is that we aggregate the single cell RNA reference profiles to get reference profiles for the major and the minor cell type. This is done via user-defined hierarchy, so we do not automatically filter how those cell types matches together. You need to put this into the algorithm. And then we learn gene weight specific to distinguishing between those major cell types, like epithelial cells and T cells. And as we assume, which is a model assumption, that we know all cell types present in a tissue, we normalize the result then to 1. And now we can use this information we got from the deconvolution of the major cell types to get a better estimate for the subcell types. First, we try to remove the contributions of other cells for one specific major cell type. This can be done by just multiplying the contributions we previously estimated for all the other cell types and subtract them from the whole bulk. I hope that this illustration now makes this clearer. For example, in the next step, we are interested in the deconvolution of the T cell subtypes. So we try to get a residual bulk that only contains the T cell RNA data and is rid of the epithelial cells and the NK cells. And on this residual bulks, which we call them, we now learn gene weights specifically to the child cell types of this respective parent cell type, which then allows the model to focus especially on genes that are necessary to distinguish between these related cell types, but aren't really necessary to distinguish between T cells and epithelial cells. So, for example, we got our residual bulk for T cells and then run the deconvolution only on this residual bulk to obtain T cell gene weights. And in the next step, we take the epithelial residual bulk, run the deconvolution to obtain epithelial gene weights. The last step, sorry, is then to use the information we obtain from the deconvolution of the major cell types and the hierarchy. Because we know that the sum of the CD4 T cell and the CD8 T cell in one single sample should not be larger or smaller than the estimated proportion of T cells. So we just normalize those bulks to sum up to the parent. As a second part of this webinar, I want to present you an example deconvolution pipeline like we also did in the article. We do this on breast cancer data. Therefore, we downloaded data from the Disco Immune Cell Atlas, which is single cell RNA sequencing data. We need this to create training and test bulks because the model needs to have data with the known ground truth proportion to learn those gene weights. The downloaded data set consists of about 200,000 cells with 34 cell types on the finest hierarchy level. As the algorithm uses raw gene count data, we only include raw gene count data. We didn't include any normalization with the training with the single cell data. We just took it as it was. Then to benchmark our own algorithm, we split the data into training and test data before doing any further processing with the data. The test data set then is put aside and not used for any training of the model, only for evaluation of the algorithm. And as the data consists of roughly about 20,000 genes, we need to reduce this to have a lower computational burden. We know that most of these genes aren't necessary to predict the cell type. So we selected the 5,000 importance genes by calculating the variance of those genes across the cell types and then use the 5,000 with the highest variance. As a last step for data pre-processing, we simulated bulk samples with a known cellular composition, which was just done by randomly picking 100 cells, adding them up, and doing this for 10,000 sample bulks. We also did it with the test single cell data to have test samples. Height then needs a hierarchy, which we created by going through literature and see how the cell types of the single cell data are related to each other. Then the model was trained on the training data, and we validated the results on test data using Pearson's correlation coefficient. First, we were, of course, interested in how our model compares to other state-of-the-art deconvolution methods. Well, the most known deconvolution method, I guess, is CyberSort, which is almost included in any paper that runs deconvolution. So, of course, we included this in our benchmark. CyberSort works with new support vector regression. Another approach to cellular deconvolution is by using Bayes inference to maximize the likelihood, which is done by Bayes prism. And the third method we benchmarked was music, which uses a weighted non-negative least squares regression approach. We use the exact same data, both for training and evaluation, as we put into height so that we can have comparable results. The benchmarking tables are pretty large because we have 3 layers, major cell types, minor cell types, subcell types, and we have 4 methods. So I only included like the summary here, and you can definitely see that for this case, height outperforms the other methods on all 3 layers. Then, of course, we were interested in how our model performs on real-world data. So we used the trained model on breast cancer samples that we downloaded from the Cancer Genome Atlas, or TCGA. And then we performed 2 example analyses. First, we associated cell types with their effect on patient survival, and then analyzed differences in the tumor microenvironment between various cancer stages. Because we didn't have enough samples, we only defined 2 cancer stages, meaning one which only includes a primary tumor but no lymph node infiltration or metastasis, and the second group is primary tumor plus either lymph node infiltration or metastasis. To perform the survival analysis, we considered each of the breast cancer subtypes that were in the TCGA bulks, namely luminal A, luminal B, triple negative breast cancer, and HER2 positive as a single cohort, and trained a Cox proportional hazards model on it using a Python package. The estimated cell type proportions then were used as covariates in the training. In total, we found 37 cell types across the cohorts that were predicted to have a statistical significant impact on survival, which means they were predicted to have an effect size and a p-value less than 0.05. And I want to briefly show you 2 examples of cells that had an impact on survival, and which also shows that we need to predict on the finest coarse layer of cell types. First, we found that CXCL13 exhausted CD8 positive T cells were associated with improved survival, which is also backed by the biological literature, as for example, Lin et al. Showed that high levels of CXCL13 promotes the formation of so-called tertiary lymphoid structures. These then seem to facilitate antigen presentation and lymphoid cell recruitment within tumors. On the contrary, we also found T-regulatory T cells, which led to a poor prognosis in triple-negative breast cancer, which we also found being backed by the literature, as Tregs are able to suppress immune activity and thus impair immune response against tumors. And now the interesting part comes. We also looked at how major T cell levels influence survival, and we weren't able to find any significant effect, which shows us that for meaningful results, we need to focus on predicting the smallest grain cell type proportions, even though those are the hardest to predict. So to summarize this talk, with the height model, we were able to predict cell type populations on multiple cell levels and are able to disentangle molecular related cell type populations. Sorry. Furthermore, we maintained consistent results when regarding different cell type levels. If you now are interested in the better detail and more details about the algorithm or in the source code, I've provided here 2 QR codes leading to the GitHub repository and to the bioinformatics publication. And as of last, I want to thank my 2 supervisors, Franziska Görtler from the University of Bergen and Michael Altenbuchinger from the University Medical Center Göttingen, and also Sarah Bolz, who helped me with all of the medical interpretation. Thank you very much.
Yeah, thanks a lot, Dennis, and a very nice talk. And yeah, now it's time for questions. So for the audience, if you have any questions, you can raise up your hand, and then I will let you talk to the speaker and just let me know. And or if you want, you could also type your questions in the chat, and then I can read it out for Dennis, and then he can try to address them. So obviously, this is a very nice talk, and I probably can start with some questions. So you mentioned that for your training data sets, you have used this breast cancer, right? There are like up to 200,000 cancer type cells.
Yeah.
My question is like you also have this kind of how 30 something cell types. So the first biology question is I have is like, are these defined cell types? Is like a consensus knowledge that is already fixed for this particular cancer,
Um,
or this because maybe this may differ, right? Biology is evolving time to time, and different people may have different definitions for different cell types. But how do you ensure the one you used for training your model is reliable, and whether these are any kind of consensus agreement on
yeah.
this kind of data you used?
Yeah. Thank you for the question. And you now mentioned a point which is a like not a problem, but which is a point with reference-based deconvolution because you need those consensus
Yes.
cell type profiles. In our case, DISCO is a atlas of many single cell RNA studies combined together, and they already had a consensus cell type annotation coming with them. So
Okay.
we didn't do any relabeling. We just took their labels because we assumed them to have thought about this. But yeah, you're right. If you use another labeling, you could probably get different results.
Okay. Yeah. So follow-up question on that. You mentioned that you use this top 5,000 variable genes for your model. So my understanding is like for single cell data, actually for most cells, to my knowledge, usually you will have a very sparse data, right? Most of them are zeros. I don't know if you are to about but you have 200,000, maybe you can find this 5,000. I mean, to my knowledge, if you have only like several thousand or 10,000 cells, you probably cannot even find 5,000 genes that can be measured or are measured in the data. So how does this really affect your algorithm? I mean, if you select fewer or more, is there any like systematic analysis on how these gene numbers affect your results, or how? I mean, also you split the data. You didn't mention how did you exactly split the data into training and the test data sets, and how does this you know processes or data processing steps affect your results?
Well, let's first talk about the thing with the genes you mentioned.
Mhm.
I did a study which we didn't include into the paper because it was an ISMB paper, so we were limited in our stuff that we could put into. But I tried two different approaches because there is biological literature which states marker genes for cell types, and I was interested in are those marker genes relevant for the deconvolution. So I created two test samples. In the first test sample, I only included marker genes but removed all the other genes, which
Mhm.
then came down to like 100 or like I don't know
Mhm.
the exact number, but like
Very
around 200,
few. Mhm.
and then run the deconvolution. And on the other side, I just removed all of the marker genes, but took in like 6 or 7 thousand genes. And we were able to see by that the marker genes are pretty important. So if you have all of the marker genes included in your set, but remove most of the other genes, you get reliable results. If you have a high number of genes, but remove the marker genes, we were still able to get good results. I
Mhm.
think if you're using a low number of genes but don't have the marker genes in it, this will lead to worse results.
Okay.
Um, then concerning the second questions about how we split it into training and test samples,
Hmm.
there are different ways how you can achieve this. First, you could, for example, split by patients, because from many studies, a patient where the single cell data stems from. So you could, for example, say we take 30% of all of the patient cells, of the cells of 30% of the patients into the test set, and the other 70% into the training sample, which is one way. But for this, it is necessary that the patients have exactly the same diagnosis. So, because otherwise it could happen that you, for example, have one breast cancer subtype only in the training data and one breast cancer subtype only in the test data. Another approach which we used for this paper was like uh, taking half of all cells of one type into the training data and the other half into the test data.
Okay. Are there any other questions from the audience? I hope there are. It seems like nobody is raising their hands or, or unmute themselves to ask questions. Maybe, sometimes some people are not in this field, but this is time to time happens. Yeah, I guess you have addressed most of my concerns, and I think that's a very nice work. And yeah, and I think we are also about the timing. I mean, the time is approaching to the end, and I think before we finish, I guess we have